Hi, my name is Irene and I'm going to present my work titled Molecular Docking Analysis for the Identification of Potential mTOR Inhibitors Obtained from Natural Products of the Peruvian Biodiversity. The mammalian target of rapamycin, or mTOR, regulates cell proliferation, autophagy, and apoptosis by participating in multiple signaling pathways in the human body. As a result, its dysregulation is implicated in various diseases such as arthritis, insulin resistance, osteoporosis, and cancer. Peru is considered one of the 15 megadiverse countries in the world for its wide genetic variety of natural products, which have evolved over time, developing chemical and pharmacological properties similar to those of drugs. The purpose of this work was evaluating 1,037 natural product structures as possible mTOR inhibitors using virtual docking screening. We started by preparing the three-dimensional structure of mTOR using the information obtained from the protein data bank. As a second step, we prepared the ligands. For this, we extracted 1,037 structures from a library of natural compounds from Peruvian biodiversity. Both the protein and the ligands were processed and used for a virtual docking screening with the AutoDock PINA program. The docking scores were analyzed as a representation of the affinity and interaction profiles between natural products and the mTOR protein structure. We selected the best ligands based on a docking score greater than -kilocalories per mole. Then, using the best ligands, we calculate their pharmacokinetic parameters and their drug-like nature. Finally, with the lickplot program, we estimate the potential intermolecular interactions of the ligands with mTOR. The result of the virtual docking screening yielded 8 ligands with better docking scores than the original mTOR ligand. We chose these 3 because they show ideal pharmacokinetic properties for good oral bioavailability. And moreover, they follow all the rules of Lipinski, Egan, and Bieber. Here we can see the clustering of the best natural compounds based on the similarity in their molecular descriptor values. Egan's boiled egg graph showed that the 3 compounds have the potential to cross the gastrointestinal tract just like the original ligand, and only 2 of them could cross the blood-brain barrier. The 2-dimensional diagrams show that the main amino acid residues that are associated with ligand binding are isoleucine, lysine, leucine, valine, tyrosine, and aspartate. These interactions are mostly London forces, which are a type of van der Waals forces. In conclusion, the structures of the natural compounds identified as GC3-120, GC3-335, and BAG-183 resemble the structure of the original mTOR ligand. These structures correspond to Streptomyces sp with antibacterial properties, Acurantes bidentata, and Aspidoderma excelsum, both with antimalarial properties. The results give us a promising future for the development of potential mTOR inhibitors and the possible cure of diseases such as cancer. We would like to extend our gratitude to Vicerrectorado de Investigación y Posgrado de la Universidad Nacional Mayor de San Marcos for the financial support and to Instituto de Investigaciones de la Amazonía Peruana for the facilities provided. We also want to thank the organization of this event for giving us the opportunity to present our work.